Assalamu alaikum everyone. I am Benramdane Zeynrb, a PhD student in organic chemistry at the University of Msila. I'm going to present the following research under the title In silico molecular docking prediction of isolated flavonoids from hyacinth with lingulata against Parkinson's disease, adenosine A2A receptor. At the beginning, we could say that Parkinson's disease is a chronic neurodegenerative disorder characterized by impairment in various motor and non-motor functions of the body. Over the years, patients diagnosed with a PD are often given dopaminergic agents, such as aldopam. Nonetheless, side effects can significantly impair the life quality for patients and limit therapeutic indications of such kind of drugs. Nowadays, phytochemicals including flavonoids were found to exhibit significant neuroprotective activities that can potentially offer effective and safe anti-Parkinsonian agents. In this regard, in vitro assessments revealed that apigenin might have an inhibitory property against the release of pro-inflammatory cytokines, whereas luteolin could inhibit the activation of microglia and protect the LPS-induced dopaminergic neuronal degeneration. In a previous phytochemical study, 10 glycosylated luteolin and apigenin were identified in the butanol extract of hyacinth with lingulata, where this later show good anti-inflammatory and antioxidant properties. Thereby, a recent investigation regarding novel approaches for therapeutic intervention in PD exposed that the adenosine A2A receptor antagonism is a promising therapeutic approach for a PD treatment. In the current study, we are aiming to predict the sort of interactions that may occur between the A2A receptor targets and the glycosylated apigenin and luteolin, and to investigate also how the type, number, and position of saccharides could affect the. So, to conduct this molecular docking study, the elected phytochemicals in Figure 1 were sketched using ChemDraw, then optimized using Avogadro software. However, the crystal structure of a human A2A receptor in complex with xanthine under PDB code 3RAY was downloaded from Protein Data Bank online site. The input PDB-QT files of both ligands and targets were created using Autodoc tools. Then the docking calculations were performed via Autodoc Vena software. Meanwhile, the 2D and 3D illustrations of ligand-receptor output interactions were generated using Discovery Studio and Chimera software, respectively. Therefore, the redocking results in Figure 3 show that the pulse at 7.2 kcal per mole was found to superimpose closely on the experimentally reported conformation of the reference ligand xanthine, with an RMSD value of 0.128 angstrom, which was accepted to validate the used protocol. Likewise, the docked iglycons apigenin and luteolin are aligned inside the active site of the target as shown in Figure 4. Table 1 displayed that all the glycosylated flavones show high binding affinities to the receptor, arranging similarly inside the active site pocket when compared to the standard xanthine, free luteolin, and apigenin. For the glycosylated apigenin groups, the presence of an additional glycosyl unit increases the affinity of the apigenin skeleton to the active site residues by involving its structure with more interactions in compound 1, 2, and 4. The 3 rings of those compounds are shared in the same hydrophobic interactions with the crucial amino acid residues, phenylalanine 168, methionine 270, and isoleucine 274. However, the glycosylation at the para-hydroxide position in compound 3 displays a remarkable ability to interact differently with amino acid residues, forming more hydrogen bonds. In the case of investigating the influence of introducing the rhamnose unit, as illustrated in Figure 6, the replacement of glycose unit by rhamnose increases the affinity of compound 7 to the receptor by forming hydrogen bonding between the K amino acids asparagine 253, methionine 270, and the hydroxyl groups of the glycose unit. But then again, the branched attachment of saccharides increases the binding affinity of the isomer 5 compared to the linear connection in compound 6. Moving to the glycosylated luteolin groups, the molecular docking study stated that when luteolin skeleton was substituted as 7 and meta-hydroxy positions with saccharides, the obtained compounds 8, 9, and 10 are efficiently docked in the active site of the target via creating numerous hydrophilic interactions with the active amino acid residues. For those compounds, the position and the number of glycosylation influence slightly on the number of interactions with the K amino acids in the active pocket. To sum up, asparagine 253 have a key role in the activity toward this target via hydrophilic interactions. Indeed, this amino acid residue also serves as a strong hydrogen bond contributor for the docked compounds 5, 7, and 10, in addition to the hydrophobic interactions with phenylalanine 168, glutamic acid 169, and methionine 270. In conclusion, these results suggest that the glycosylated apigenin and luteolin may exhibit inhibitory effect toward the adenosine A2A receptor, mainly the selected compounds 7, 10, and 5, where they possess the ability to fit well into the binding pocket through forming hydrophilic and hydrophobic interactions with the crucial amino acid residues. This theoretical study could provide precious insight for designing a lead compound for Parkinson's disease, starting from the base structure of glycosylated apigenin and luteolin by targeting the adenosine A2A receptor. And thank you for your attention.